Morning, everyone. Thank you, Mike. Thank you to the organisers very much for this invitation. So sticking with the esophagus, but changing focus a little bit to adenocarcinoma and telling you about our particular experience in the UK. So this is a map you're getting quite familiar with this morning. But we've been focusing so far on squamous cell carcinoma, and you can see that the UK is highlighted here also as a red high incidence area. But in the UK, that's due predominantly to adenocarcinoma. And just to make sure everybody's completely clear about the difference, anatomically, the adenocarcinomas occur generally in the lower third of the esophagus, particularly at the junction with the stomach. And actually, it can be quite difficult sometimes to tell whether an adenocarcinoma occurring right here at the junction is really predominantly esophageal or gastric, and that's something we might want to discuss in more detail vis-a-vis Reza's comments just now about gastric cardia and whether we should include that. We know that the adenocarcinoma is more common in men than women. We have a rather crude understanding of the risk factors, but chronic reflux seems to be very important. Obesity may also contribute, and this has become the predominant subtype in Western countries. And just to highlight that, here's the data on men and women from the UK between 1971 to 2013, and you can see this really dramatic increase in adenocarcinoma amongst men in the UK. And if we look on an arithmetic and log scale, this is white males, this is the SEER data of increasing incidence of esophageal adenocarcinoma, so this is US data. And then when we break that down, white men, white females, black females, you can see that the biggest burden of disease is in white males. And this is our, as I say, our somewhat crude understanding of the risk factors based on the epidemiology that we know. But this is where the Mutographs project might really add value to understanding what some of the mechanisms are behind these processes, because for a long time we've known that acid and bile epidemiologically seem to be important, but we really don't understand what the fundamental mechanisms are by which they induce adenocarcinoma. What we also understand about this condition is something about the pathogenesis by which we go from a normal squamocolumnar junction, this is the endoscopic appearance and the matched histopathological appearance, to this pre-malignant metaplastic state where the lower esophagus is replaced by a columnar epithelium, as shown here. It's rather a mosaic epithelium with some characteristics of gastric epithelium and some of small intestinal epithelium with these goblet cells, which are pathognomonic. And in a small proportion of patients, that can gradually progress through dysplastic stages to adenocarcinoma. The majority of patients with Barrett's esophagus will not develop adenocarcinoma, but the risk increases with these dysplastic states. And this is clinically very important because the survival drops dramatically by the time you have invasive disease. And now we have really superb endoscopic therapies to cure patients who are identified at the dysplastic stage. So there's been a big effort to perform sequencing, particularly in the cancer, but also to some degree in the pre-cancer, by the TCGA and the ICGC. The data I'm going to share with you today is our ICGC whole genome sequencing data funded by Cancer Research UK, and we've now submitted most of our cases, nearly 200, to the Mutographs project that we'll be able to add to this experience shortly. This is just to highlight the fundamental difference between squamous and adenocarcinomas of the esophagus. So very often in the literature, these cancer types are actually lumped together. In clinical trials, we often lump them together, but these are fundamentally different cancer types, both histopathologically, epidemiologically, and whatever level you look at with regards to the genomic, transcriptomic, or epigenomic profile. So these really are different diseases. But as I say, the interaction between what's happening in the esophagus and the stomach at the junction is rather more complex. This again is TCGA data looking at cases of the esophagus, cases at the junction, cases of the stomach, and trying to group them together somewhat based on their genomic profiles. And to highlight here this purple subgroup, which is the cases mainly occurring at the junction, which they've described here as chromosomally unstable. As you move towards the distal stomach, there are some differences with more predominance of MSI and Epstein-Barr virus-associated tumours that we really don't see with the tumours occurring more at the junction. So this was our first pass from the ICGC data of 129 cases, really just to highlight what a heterogeneous cancer it is genomically at every level, pointing out here the recurrent point mutations, and loss of P53 mutation and loss of P53 is by far and away the most common mutation that we see in this cancer subtype, and these mutations tend to occur rather early. After that, when we're looking at point mutations and indels, there's a very long tail of low prevalence in a whole number of genes. There's a lot of fragile sites that are affected, and when we look at the copy number alterations and rearrangements, we start to see some rather more recurrent patterns, and it's a very complex tumor in terms of the copy number variation and structural variation, as I'll also highlight later. But what got us interested in the signatures early on is that there is some homogeneity in the signatures when we look at these cancers. And this was our first looking at the prior classification of the cosmic signature types, finding here 6 most common signatures. And when we start to look at the most dominant signature in each case, then we see 3 main subgroups from this 129 cases, which we described here as DDR-impaired, mutagenic, and this C2A dominant transitions. The mutagenic subgroup, so called because it has a higher mutational burden and is really dominated by signature 17. We were interested in the DDR group with this BRCA-like mutation because germline mutations in BRCA are extremely rare in this subgroup. And when we looked at the DNA damage repair pathways using this pathway analysis, it's interesting to see that in this DDR-impaired subgroup, this is really coming from defects in homologous recombination, particularly including genes like RAD51, REC-L, not necessarily BRCA. So we've got a bigger cohort now to examine, and of course the methods for looking at whole genome data have improved significantly, and so that's enabled us to start to get a better handle on what's going on. And so what I'm going to show you here is some new data on 413 whole genome sequences of esophageal adenocarcinoma, including tumors around the junction, based on the SIG profiler database. And now we start to see a number of more signatures, and when we perform clustering analysis, now we can see 4 main subgroups, the mutagenic and the DDR that we saw before, but now the other, what we call the C2A dominant group, is divided up with this ROS and MMR signature. So the additional signatures are 30, basic excision repair, also platinum-based therapy, because we have included in here chemotherapy-treated cases. Also MMR. So if we just look at these in a little bit more detail, so the MMR subgroup accounts for just 4% of this cohort, and here we're seeing the dominant recognized MMR signatures 20, 26, and 44 shown here in the lime green. The mutagenic signature, which remains the commonest subgroup, 48%, and this is really dominated by signature 17, shown here in the purple. And there are 2 slightly different forms of signature 17, which are called here A and B. Here's the DDR signature that we also saw before, 21%, with BRCA, aging signature. The APOBEC signature is also a small contribution in this group. And I think this SBS3 signature is quite interesting as it's associated with indels in microhomology regions. Here's a ROS subgroup, which we couldn't really distinguish before until we had these sorts of numbers. So this corresponds to what we called the C2A CCT subtype in the previous classification. And here we're seeing more signature 18, which is associated with defective mute enzyme base excision repair. This is just showing you the mutation load across the subtype, because I said that in our previous analysis we'd shown that what we called this signature 17 mutagenic group had a higher mutation load when we consider per megabase. This continues to be the case compared to these 2 groups, and as expected, I guess the MMR group has the highest mutation load. But what about looking back in the history of this disease at the Barretts? Because I think this is a very nice opportunity we have in this cancer type to study the pre-cancer, because we monitor these patients repeatedly over time. And so this is around, I think, 140 cases of Barrett's esophagus grouped into different stages. This is a little bit complicated, but these are non-dysplastic Barrett's esophagus here. And we've divided these into patients that have never progressed versus patients that did eventually progress, that we've called here pre-progressors, patients with low-grade dysplasia, high-grade dysplasia, intramucosal carcinoma. And then we've put these separately. These are cases where we've got a cancer and associated pre-cancer, pre-malignant Barrett's tissue next to the cancer, but they've only presented at the time they had a cancer. And I think, the sample taken from that case may be non-dysplastic, it may be dysplastic, it's just, due to somewhat to sampling bias. So we've considered those separately. The point here, I think, is that you can see that at first glance, there's not an obvious difference in the spectrum of mutational signatures as you move across these stages. And we still see many of the signatures we see in the cancers, and it's still dominated by this signature 17A and B, as well as SBS 40. I don't know if we're going to hear more about signature 17 perhaps later on, but I think this is a very interesting signature that we still don't fully understand, but some interesting things are becoming clear about it in terms of replication strand bias, its relationship with the nucleosome. But it's recently 2 papers in bioRxiv about 5FU treatment, which induces a very similar appearance to signature 17, shown here in model organisms. This is organoids, 5FU treated organoids, and this is 5FU in Leishmania from Nuria Lopez-Vigaz. And this really recapitulates signature 17. I did therefore have a look at our cases in relation to 5FU treatment. So this is signature 17A, B, and grouping them both together. This is Barrett's esophagus. So these patients are pre-cancer, they've never had any 5FU treatment. These are treatment naive, no chemotherapy, and these are treated cases. And you can see that we see signature 17A and B across all of these groups. So I think that tells us that, this can be induced by exogenous processes as well as presumably as endogenous processes, and it perhaps gives us a window into how to go about exploring the mechanism really underlying this. Just wanted to briefly touch on structural variation because this is such a rearranged tumor type. And this is just grouping our tumors together to show you that we have a lot of deletions, translocations, duplications, and inversions. And we can start to look at patterns in these as defined by Serena in breast cancer. And when we look at whether these rearrangements, according to whether they're clustered or non-clustered across the genome, and we look at the different types, deletion, for example, we can see here that there are different patterns that emerge, and we can then define these as whether they're signature 1, 2, 3, or 4, whether they're clustered or non-clustered. The point is here there are patterns that emerge, and these are rather similar to breast cancers, but we don't observe the tandem duplication that they do, which is well known to be associated with homologous recombination defects, and that's not surprising because that's not something we really see much of in this cancer type. And if we start to compare our SV clusters with the SNV subtypes, just a couple of things worth drawing out here. So if we look at this group of cases here, this is the DDR SNV subtype, which are more common in relation to tandem duplications as defined by the SV signature. And we can see that these tumors are characterized by rather complex rearrangements. If we look at this spike here, this is signature 4, and it's clustered. We can see that this group also has a high proportion of fragile sites, mobile elements associated with them. I think model systems are really useful to try and perturb the system and understand what might be driving some of these signatures. And of course, all these model systems have their pros and their cons. We've been working with Matthew Garnett and colleagues, Hayley Francis at the Sanger, to develop organoids from the same primary cases that we're using, or a proportion of them, for the ICGC whole genome sequencing project. And we can then use these to do clonal evolution studies, for example, and look at how they alter over time. And there is clonal evolution that occurs, although the signatures, when you don't perturb the system, seem to stay somewhat stable. But then I think this gives us a nice paradigm in which we can alter various things in the system, exogenously or genetically, to see what happens. And we've used this system to do some in vitro therapy testing to see if we can test some hypotheses about how the mutational signature might inform us as to therapeutic susceptibility. And this was work a few years ago based on that initial analysis with the signatures of 129 cases, coming up with some hypotheses based on the in vitro data about what therapy targets you might consider. For example, in the mutagenic group, thinking about immunotherapy perhaps, or CHEK1/WEE1 inhibition, and we've been starting to develop some clinical trials around these hypotheses. So just to conclude, I think that esophageal adenocarcinoma can be clustered based on their base substitution, sorry for the typo there, signature profile, which is quite useful when it's such a heterogeneous cancer. New signatures are emerging as we've got bigger numbers and new methods. Signature 17 is very intriguing. It's the most prevalent. It's seen in the context of exogenous 5FU, but presumably also endogenous processes are resulting in this signature. And we see it early on in the pathogenesis in the context of Barrett's. This is a highly rearranged cancer type, so I think we need to also think about what's happening there and the causation. And I hope that the Mutograph project will, especially with the very good clinical annotation and questionnaire data, will really help to add some further insights into this disease. So I need to thank the team members who have done the work, particularly Alvin Ng, postdoc, Sujat, and Shuganesh, who have done the work that I presented today. Thank you very much.